So thank you so much for the invitation. So today my topic is mitochondrial function and aging implication for among people with HIV. So I really appreciate that Dr. Du could just bring up this excellent review paper on hallmarks of aging. So as you can see that hallmarks of aging are really interrelated and interconnected with each other, and these new proposed 12 hallmarks are really satisfying 3 premises, including the alteration over the aging process. The second premises is how they could be possible to modify during the lab and observe that in the intervention. And the very last thing is what we can do with these aging markers. Like they are all satisfy in fact an element that we can potentially modify of these factors. So that brings it to be a really exciting topic. So I also want to bring back this figure proposed a couple years ago in an NIH workshop on HIV and aging. As you can see, people with HIV is really susceptible to multiple factors across their exposure to HIV, including like antiretroviral therapy, the viral reservoir co-infection with hepatitis C or hepatitis B, as well as HPV and such. And then they also have numbers of lifestyle and environmental stressor that can constantly impacting their aging process. So all of that are mediated through these immunosenescent mitochondrial damage, epigenetics, and the stem cell exhaustion process. These are the 4 primary mediators that originally we proposed about. So as a molecular epidemiology, my interest is really starting from my postdoc at Hopkins and started to looking at mitochondria. So as you can see, mitochondria is really existing in almost all of the eukaryotic cells, and then it contains a much smaller genome, including only 36 genes, and then including 13 genes used in the cellular energy production. Mitochondria has always been known as the powerhouse of the cell, producing ATP and through the oxidative phosphorylation. And then mitochondrial DNA variation has been studied a lot in the past decades or so, and it has been linking to the mitochondrial function and multiple aging-related disease. So since I started my postdoc, and I started to be introducing to the topic to evaluating mitochondrial haplogroup and how that may be related to multiple aging-related outcome among people with and without HIV. I'm doing that because I have the fortunate access to lots of the genotyping data in the MaxWise cohort as well as the Alive cohort in at Hopkins. And then, so mitochondrial haplogroup is generating by a combination of SNPs in the mitochondrial genome, and then it usually represents some accumulation of the mutation through human migration process. And as you can see that human migration across the world can sometimes expose to different kind of environmental stressor and environment, latitude and such. And so all of these leading to a number of adaptation throughout the human evolution. So, so ancestry, all of these components may be having some functional implication among our human. So with all of these resources I got access to, we started with a study to look at the mitochondrial genetic variation and how that linked to the physical function among MAX cohort, which is a cohort of people who men have sex with men, and we including about 450 men MSM population, and then with half of them all of them are HIV positive. And then among them, even with such a small sample size, we actually observed that one of the mitochondrial haplogroup, J, European haplogroup, that is actually predicted to the physical function decline among people with HIV. So that's really interesting to me. And then a further investigation in mitochondrial haplogroup and other aging-related disease such as diabetes also revealed that, for example, this study, we observed that in the WISE cohort, which is a cohort of women with high risk of HIV infection, and then we observed that one of the African mitochondrial haplogroup is associated with decline incident diabetic risk, and then another haplogroup L3 is associated with increased incident of diabetes. And we also dive into look into further whether or not these haplogroup can differentiate people with their beta cell function. And indeed, we did observe that mitochondrial DNA haplogroup L3 is associated with a faster decline in beta cell function, and L2 is associated with a slower beta cell function decline. So with all of these knowledge, we turn to, and then one of the interesting observations we found from the previous WISE investigation is that all of these association is really only observed among people with HIV, but it's absent among people without HIV. So that prompts us to think that, is there any special characteristic among people with HIV could pose some gene and environment interaction, and then leading to such a different outcomes. So we look into the max cohort again, and then we're trying to define that whether or not among people with HIV, whether or not the haplogroup and antiretroviral therapy exposure, whether or not they have independently and aggregately impacting the diabetes risk. So in this max cohort, we stratify our population into the European population and the African population, and then we evaluate the common haplogroup within the two groups and how that interact with the antiretroviral therapy. And again, we only observe that number of haplogroups, such as the European haplogroup UK, as well as the African haplogroup L3 is associated with a significant increased risk of faster time to diabetic. And then although we did not directly observe an interaction between the antiretroviral therapy, namely those mitochondrial toxic antiretro ART, early generation NRTI, we did observe the group exposed to both L3 haplogroup as well as the D drug are the group with the fastest time to develop incident diabetes, as well as highest risk of developing these conditions. So with all of that, we started to think that what is the clinical implication for these haplogroups? So as you can see, this is really nuances, differences in the mitochondrial genome. But then, if you think about that, fast forward, all of these observation and data may ultimately contributing to some somewhat robust algorithm in the future with the computer science advancement. We might be contributing collectively to the precision medicine or risk stratification in long run. So the other question to us is, we do know that some of the common way of studying mitochondrial genetic variation, one way is, as I talked a lot about just now, the haplogroup, which we do aware there are limitations to that because this is largely driven by the ancestry, and then also can be significantly confounded by the ancestry components of it. So we also know that haplogroup is not directly functionally implicated or relevant because it's really just a proxy of the function and human adaptation to the environment over time. So what can we do more than just a proxy for that? And the other way of studying the mitochondrial genetic variation is single variant tests. So compared to the nuclear, mitochondrial genetic is more unstable and have more mutation across the lifespan. And then it also can be haploid and then with multiple variants occur. So that poses a lot of challenge in the previous investigation in the mitochondrial genome and require a much higher multiple testing burden. So all of these collectively is become a challenge in our previous investigation. So I want to bring up one of the recent study that working with our collaborating lab is this study is using a very different approach, trying to studying the mitochondrial genome collectively using the functionally informed mitochondrial haplotype. So here is using GWAS data in the UK Biobank, and then we started to the haplotype. We started to generate the haplotype against all of the 41 mitochondrial copy number relevant traits, as well as the mitochondrial copy number. And I will talk about mitochondrial copy number later. But basically, the idea is that mitochondrial copy number is a marker for mitochondrial biogenesis. And then the trait that relevant to copy number may also indicating your mitochondrial function and efficiency. So after the regression, we do the fine mapping and identify 7 credible sets of mitochondrial SNPs listed here in this figure. And then we, through that, we also identify 6 distinct SNPs capturing 8 common haplotypes in the UK Biobank. And then our investigation also further look into that and trying to see the non-trauma or non-accident related mortality and how that related to these mitochondrial haplotype. And indeed, we did observe that these haplotypes are significantly associated with non-accident related trauma in the population. So another characteristic of mitochondrial genetic is that mitochondria is quite different in terms of the genetic variation. There is also a feature of homeoplasmy and heteroplasmy occur among in mitochondria. So simply speaking, homeoplasmy is usually represented an inherited variation in mitochondria. And usually, as you can imagine, since this is an inherent variation, it usually carry out in larger scale and then pose severe manifestation among people. So that lead to disease such as the liver hereditary optic neuropathy and then the mitochondrial myopathy such. These are rare disease, but it does unfortunately occur in the general population. But the other characteristic of mitochondria, which is heteroplasmy, is actually much common than the homeoplasmy. So what it suggests is the somatic variation and mutation across lifespan. Usually, some copy of the mitochondrial DNA have mutation, and then the level of that and the specific SNPs that has been mutated are usually represented as some functional implication and disease-related trait. So just to put it in context, about 30% of people actually have more than one heteroplasmy across their lifespan, and then accumulation of these mitochondrial heteroplasmy can occur during the aging process as well as when we expose to environmental stressor. And this study here I show is also another demonstration of how these heteroplasmy are accumulated. So for example, in these, the panel A here is really just an average heteroplasmy level over the age spans and how that accumulated among all of the marker the investigator study. And then on the second panel B, which is a randomly selected Jing, and then as you can see that we also can observe that the age-related heteroplasmy in this particular gene mutation, but then there is always exception, right? About 10% of the gene actually has a reverse association, suggesting the level of heteroplasmy is not accumulated over age, but then reduced. So that poses a lot of complexity on studying the heteroplasmy. And so here is another study that done in our collaborating lab. So using the whole genome sequencing data in the UK biobanks, we characterized the mitochondrial heteroplasmy, and then we created a score of mitochondrial harmful or disease-related heteroplasmy, and then we actually noticed that the characteristic of that score, the characteristic of heteroplasmy, actually predicted almost all cause and then cause-specific mortality, as we show in this right panel. So that suggests that there are a lot of potential in terms of aging research and in terms of how we can understand better mitochondrial genome and mitochondrial functionally relevant genome especially, and how that can apply in our ongoing aging research. So, but the other more relevant question to us is, can we do a more direct assessment of mitochondrial function? And that's always the question. So, as many of you already familiar with this figure from maybe high school textbook, this is really the cellular molecular mechanism of mitochondrial function and how that interact and contributing to the Krebs cycle. So basically, mitochondrial function can usually be measured such as mitochondrial bioenergetic, mitochondrial calcium signaling, the morphology, the mitochondrial fission and fusion. However, not all of these functions are very easy or readily available to measure for a researcher like me using large-scale population cohort observational study setting. So one of the marker that's emerging, that's readily available and relatively cheap and non-invasive is actually mitochondrial copy number. So the idea behind is because mitochondria usually present hundreds to 1,000 copies in each of the cell in relative to nuclear DNA, we can try to see how many mitochondrial copy number, which use that as a marker for mitochondrial bioenergetic and biogenesis in cell as a proxy of mitochondrial function. So this one study published, 2 decades ago by now, is to use this mitochondrial copy number as a marker to look at the NRTI toxicity and how that impact mitochondrial function in people with HIV. So as you can see, that early generation of NRTI reduced the mitochondrial level among people with HIV. But how do these change impact aging and age-related comorbidity over time is still unknown. Also, whether or not there is any residual toxicity among people who exposed to these early generation of NRTA are really might be a continued question for us studying aging among people with HIV. So do we observe a similar impact in the post-antiretroviral therapy era is another question. So here we initiated a study in the AIDS linked to the antiretroviral exposure cohort, the ALIVE cohort, which is a study among people who inject drug. And we study the mitochondrial copy number level repeatedly over time and trying to see how that is related to people with HIV infection as well as their HIV disease severity. So this study we published a couple years ago showed that even in the post-antiretroviral therapy era, post-2005 and 2015, we still observed that people with HIV on the very left panel here shows that people with HIV are still with a significantly lower mitochondrial DNA copy number compared to people without HIV. And the good news is that if our participants who are engaged in antiretroviral therapy, show that in the middle panel, we actually see that their copy number are almost close to people without HIV. So that's really good news. But as you might be aware, people who inject drug with HIV really a challenging group in terms of their care continues. So we still observe a fairly high level of low-grade viral load among these patients. So we do see the significant effect driven by the low CD4 cell count and high HIV viral load among this population. And the next question we have is whether or not these kind of copy number are continue to decline over time. And indeed, we can see that among people with HIV, as you can see that from the lower two bar here on the left are all of the people with HIV, and then you can see that there is the copy number are actually significantly declined in a much faster pace among our people without HIV control. So that's just that the mitochondrial function may be continue to decline among people with HIV. And we also interested in what is the functional implication or long-term disease-related implication for these mitochondrial copy number marker. And within our cohort, we actually observed that among people with HIV and have low mitochondrial DNA copy number, we observed that they have a significantly higher risk of death all-cause mortality, as well as much faster time to death within our observation period as that compared to people without HIV. So that's just that potential, the mitochondrial function and biogenesis really might have a very important aging-related implication among people aging with HIV. And the other study we dive in further is trying to look at how the copy number is associated with incident chronic kidney disease among the same population. And we also similarly observed that mitochondrial DNA low mitochondrial DNA copy number is significantly associated with faster time to incident chronic kidney disease. And then we also observed that among those people with lower mitochondrial DNA copy number, we see a significantly faster kidney function decline among our population. So these are really stress the importance of studying mitochondrial function. And then we also expanded our investigation and further study to using other approach to evaluating mitochondrial function. Here in one of the NIH supplement that we just got, we are studying how the cell type specific senescent marker and how that is interactive with mitochondrial function, as well as how that may be having a functional implication to frailty and physical function in our cohort. So all of these lead to another question: can we really reverse these mitochondrial function among people with HIV? So we know that from the general population, exercise is really good for everything, right? But do we have a direct evidence that that will have a direct impact on mitochondrial function? So here we have one of the investigations through one of our amazing mentor, Christine here, observed that in a previous study that only a very small sample size of nearly 30 participants, we see that exercise intervention actually have a direct impact to multiple epigenetic marker in muscle. And what is interesting to me is that one of the marker, the biosynthesis marker is directly related to mitochondrial function. And we observed that very significant impact on the mitochondrial relevant epigenetic change in muscle, especially in people without HIV. Unfortunately, we do see the blunted effect among people with HIV, but that might pose to another question that what is the best exercise approach among people with HIV? So with our further investigation, we try to see if high intensity interval training, the HIIT here, might be impacting, giving a better improvement among people aging with HIV. So with that, I'd like to closing out my talk with this is my K framework. So as you can see, I'm very much interested in evaluating how HIV may be impacting multiple markers, hallmarks of aging, including epigenetic as well as mitochondrial function, and how that as a systematically impacting our aging and health over time. And also, I'm very much interested in how this exercise intervention may be potentially changing or reversing the mitochondrial damage in systematically, not just in one cell or the tissue, the muscle tissue. So with that, I would like to thank all of my amazing collaborators, mentors, and as well as the generous funders from supporting my K1 Development Award, as well as the NIA supplement to support our further investigation in the mitochondrial function and immune cell. So I really appreciated your attention, and thank you so much.